So good afternoon. Welcome. Hi. Thanks, Jeff. Welcome at our joint seminar, Botanical and Zoological. And this is a big honor to have such a guest here as Marten Minter is. Marten is famous ecologist, really. He came from University in Halle. Now he works in the iDiv Center in Leipzig. And his specialization is not just only biological invasions, but he studies also phylogenetic diversity, functional diversity of communities. He was one of the first one who showed that the biota over the whole world is homogenized and that the saturation point does not exist. So he really is author of very influential studies. He has studies in all the top-ranked journals you can imagine, from Nature to Science to PNAS, Trends in Evolution Ecology. I don't know what else should I mention, but so really it's an honor to have him here, and we are really looking forward for your talk.
Hello, everyone. I hope you can hear me. So you can see the long title, so I'm not speaking only about science mostly, but I also introduce a little bit the place I'm working with, for, at, whatever, it's iDiv, because it's a nice place, and I was told, please introduce a little bit this place. And sorry, I skipped the policy relevance part. I'm happy to talk about policy relevant indicators, because we just got recently some interesting results with a postdoc of mine, and I involved, and I put them into the talk. And this talk, although I have fewer publications, is never possible without many people. So I'm here on behalf, basically, on a couple of people, on postdocs I had the pleasure and still have the pleasure to work with, and a group around a database I co-founded quite some years ago. This is mainly the people I work with closely. These are people who work with GLONASS, which is a database. I, with few people, Petr Pišek, for example, you might know, founded 15 years or something ago, and these are the people who very closely work with this database. Many of the results I'm talking today are based on data and using this data. So I'm just a tiny part of it. I just have the pleasure to be invited here and to talk about this. Anyone else could do this. So briefly introducing the database, because I think, and I heard already it's used here, it's a great resource for plant invasions. So I'm an invasion guy. It's a database project, I said 12 years ago. Okay, I should update this. And GLONASS stands for Global Naturalized Alien Floras. So it's a database on usually regional occurrence data. It's based mostly on countries, but also on sub-country regions. It's a living database. We just published the 2.0 version, and it started 2015 with a paper, and you can find all the data of the database at our website, and this one I think should lead you to the data paper, or at least to the Synodo. The data paper is, I think, still under revision. Yeah, that's what I said. So it's a living database with a lot of data in there. It covers a lot of places. Okay, this is still, we already have a couple of papers using GLONASS, and it gained some attraction and some citations. So it's used. Please feel free to use it as well. What does GLONASS offers in terms of why am I working since over 10 years with a database? It covers over 1,300 regions in the world. So as I said, of course, there are not so many countries in the world. Yeah, we didn't make them up. It's also sub-country level, etc. Islands, a lot of islands. We also cover quite a lot of islands. And this is the number you can maybe memorize if you are into invasions. At the moment, GLONASS covers roughly 16,000 non-native plants, vascular plants. And you see here some data on infraspecific, subspecies, and so on. And we compiled this database from over 200 sources. And some other information is, of course, it's taxonomically harmonized. You can imagine this is always a pain. We also roughly have the information where these species come from on a quite rough level, tetraic continents, and we also gave those data more or less subjective completeness score. So how complete is the number, or is the knowledge? We don't have only numbers, we have the names. Is the list of non-native species for, let's say, Bulgaria. And we, of course, also keep the original sources. This is the first figure you see, which might be interesting. You see here in green, also, I don't know why green now. It's you see as lighter green as more non-native vascular plant species. I try to only say non-native species, not always vascular plants. And you can see there are some hotspots, the typical ones, you know, here in Europe, US, South Africa, some places in Australia. And you can see how variable this information is between one non-native plant species and over 2,500. There are some places which have more established, and that's only established non-native plants. There are some places which have more non-native established plants than natives, usually islands. And then we also lack some regions which are, I would say, the typical data gaps in many of these global studies. Yeah, we put quite some effort also to get Russian data. Nowadays, it's even more tricky. And also in Central Asia, sometimes these data just not exist. Yeah, they're also species rich, and there's very little funding for this type of research. And 16,000 species are roughly 4%. You can argue about that. I'm happy to argue about the numbers. It's roughly 4% of the global flora is non-native somewhere, okay? What else? Just some description of the data, and then I come to more interesting results. You see here the number of taxa and the typical suspects in terms of families. Yeah, we have almost 2,000 Asteraceae species in our database, and that is records. So one record is one plant per region. Yeah, and it's a bit different here, but there are no surprises, I would say. Okay. But I'm just saying, before you ask nasty questions, I'm a zoologist. Yeah. Okay. And not a hardcore botanist, so please bear with me. And as you know, I don't know how many people know that, they're also bachelor students, so you should know, or maybe you know after my talk, invasions are among the top 5 drivers of global change. Yeah? So that's why it's important to understand, to evaluate, and also to do research with biological invasions to understand them. Here come some results we did with GLONAV data and with other data over the years in a very fast manner. So for example, what we found, there is a database on first records. So when a species occurred for the first time in a region as non-native across all taxa. I mean, all taxa always means which data you find, yeah? And these are always the typical suspects, so usually not insects. We don't see in the saturation of no new species introductions. Yeah? You see them growing over time. You can now argue that this is a trend, not harmonized or controlled for recorder bias. True, but it's a global trend, which even if you control, I'm sure it holds true. Why it's going down, I guess it's just a data lack of finding these very recent data, okay? So no saturation of species introduction, although we have, since a few decades, a lot of management practices in place. There are some reasons for that curve. We can discuss that. Then many islands harbor more naturalized alien plants than native plants. Ignore that here. I thought I have done this. So many islands have more alien species than native plants. For example, Madeira, one example, more non-native plants than aliens, also called the Garden Island. And here you see either the relative richness of naturalized plants versus natives, or here you see it as ratio. And you can see that there are some places which are super red, yeah? And that means they have more naturalized than native species. And that happens usually on islands, yeah? Rarely also on some mainland regions, and then usually in those which are not so species rich. There's also some reasons for that. It's always recorder bias, history, blah, blah, blah. But that's what we have right now in our data, okay? Northern hemispheric continents are biggest donors of alien plants. This is a nice graph. I can also use this one. How to read that one? You have here, for example, North America as continent. This is with species which go from North America to somewhere, and these arrows here are the species which North America gained. It's a donor and recipient, and you can see here, maybe you can't, but the numbers are exactly what I said, that northern hemispheric continents are the biggest donors And it's changing slowly, but it's changing. Also some other regions taking up speed, mainly with globalization and also with trade. With not trade, but trade. And this is a pattern we were the first showing because we know where the species are from and where they are going to. This sometimes also people ask, what is the top invader? Now I'm showing some plant, and it's certainly boring for some of you. So these are the top 3 global invaders in our database. It's Zonchius oleariaceus. That's hopefully the right Czech name. It occurs in over 400 of our GLONASS regions. Also, no surprise, Ricinus, native to Africa. It occurs also in quite many regions. And not another surprise, Oxalis. I'm sure you also have it in many places here. And the origin here is from cultivation, which is also tricky to know. But in this case, I think the researchers are sure about this. And now few results which I found interesting, more recent results. As I said, we did quite some stuff in the last 12 years, and it doesn't fit even in an hour, also because I actually want to talk about a few other things. So what we looked also into, well, why do we see certain alien species in certain regions? And you already have seen this flow diagram. Why do North American species going into certain regions? And one of these historical reasons for that is our colonial history in the world. I mean, our means none of us was born there. But you might remember that, for example, the British Empire here, you see the British Empire. This should make or depict links between places in the former British Empire. Yeah, for example, from Britain to US. And so it's a bit messy here. And here are some names of some ports in the British Empire. And I was just co-author here, Bernd Lenzner in Vienna. He collected over several years these data for a couple of empires. So the biggest one, the Spanish, the Portuguese, the Dutch. I just learned there was no Czech Empire, which is good. But you can imagine that many hundreds of years of trade, and exchange of people and of goods, of course, also has an imprint on flora and fauna. And whoever is into invasion ecology knows that people transported, wanted to live in New Zealand and it should look like home, and they were very successful. And what we did, we analyzed these data, we combined them, we did some similarity analysis and looked for, do we see similarities, higher similarities within empires in the floristic composition than outside of empires, or then compared to just random examples of these regions. And indeed, the compositional similarity is higher and expected than by chance among regions within the same empire. So we still see today, it's also not a big surprise. I guess many people would say, yeah, I know that, or I would have expected that, but you know how science is. You have to prove it with data. And we found that there's a colonial imprint in the floristic similarity between the regions. And it also increased over time as longer those regions belong to the same empire. So quite nice study, and it used also different data than the usual suspects, phylogenetic, functional diversity, blah, blah, blah. This was a bit more interesting in terms of using some historic data. Right, and there's also, you can also see that some have an even higher similarity if they were historically or economically or strategically more important. There were some harbors which were very important because they maybe were in the middle between North America and Europe. There are the Azores, for example, and some of these islands are heavily invaded also because everyone stopped there and then all of a sudden brought their seeds and whatnot. Okay, and that's what I exchanged just yesterday for the policy relevance. Happy to talk about policy relevance, even usually happier than talking about basic science. This is the work of a postdoc in my lab, not of mine. It's not my postdoc. She's a grown-up. And it's about the floristic realms, global floristic realms. So maybe I'm asking, who knows Wallace realms? So the teachers, maybe also the teachers. Who knows the zoological Wallace realms? Please raise your hand. Don't be shy. Oh, that's botanists. No, I'm joking. So there are zoological realms where Wallace came up with, and with his knowledge 200 years ago, said, because I know that there are species which are very distinct, this is an own kingdom, realm, however, and this exists also for plants. And in this case, it was this Armenian guy, Armen Tartarian. Yeah, this is Liron, and my lab is only Liron and me. And this was Armen Tartarian, you might have heard. He did the same for plants. So he was, it was not 200 years ago, it was maybe 50, 60 years ago, but still not a globalized world where you can't, you know, finding everything on Google. And he came up with realms, he called them, and sub-realms, floristic regions. And what Liron and I thought about is, because now we know quite good where species on a macroecological spatial scale occur, we can also calculate, quantify these realms with the knowledge of the existence, of the occurrence of species. And the idea was, that was not so new. People did this already with more or less good data. You can always argue about the quality of the data, but our idea was to see what is the imprint of the non-native species. 4% of the global flora are transported across the planet, and is these realms, if we see them, do they change due to the transport of non-native species? And Stenka said this already. I'm looking into homogenization aspects since my PhD, which is unfortunately 3-4 years ago. And this is basically a similar approach, just a bit better. Yeah. And what Armen Tartarian actually found, or not found, but depicted in his work, were 6 kingdoms, 8 sub-kingdoms, and 35 floristic regions. It's not so important. The second part of my talk is about a work I did with my former postdoc, who is now a professor in Grenoble, assistant professor in Grenoble, and is very happy there, which makes me also happy on the one hand, but also we never finished this story here. It's about tropical invasions. So the idea is there's a hypothesis on which you can test everywhere. It's not a invasion pure hypothesis, but invasion ecologists adapted that if you have a more packed niche space, a more packed functional space, then there might be less space for new incoming species. Yeah, it's a sub-hypothesis of stability, whatever. And this is just a starting picture from Azores, which is hell and heaven for any invasion ecologist. You see here 5 of the 10 most invasive species on the planet fighting for light. Yeah, that was, this island was just, as I said, hell and heaven. Hell and heaven. It's really strange. So what did we do here? Again, it's macroecology. So I'm a macroecologist, okay? You have to just accept that. So the idea is, where did we start? We said, okay, we see generally fewer species invasions in the tropical regions. I showed you the map of the plants. There were lesser invasions in the tropics. And you can argue about why this is. I'm also doing this a lot. And the hypothesis we wanted to test it is very closely related to the ecosystem stability hypothesis. I'm sure there are also 10 other names for it. So in a short run, it's more biodiversity, means more stable, more resistant towards environmental changes. And tropics have fewer alien species. They are more resistant against invasions because they are more species rich. It's one way to see it. And we also try to look at the functional version of it, a more packed niche space or multidimensional functional space. Coming to that leads to higher competition and less ecological space for new species. Okay. And another reading of it, successful non-native alien species are functionally different to fill an open niche. Yeah? I hope that's understandable. And that's maybe a depiction of that. We have, let's say we look now in a multidimensional trait space, trait dimension 1 and 2, and you have these crosses are species, and here you have alien species are the orange ones, and you could think about that there's a functional trait space spanned with the trait states and values of these native species, the more bluish ones, and then you have new species, and you could analyze where they are in this functional trait space. A typical question, I'm sure hundreds of people ask that for whatever reasons. Yeah? And we wanted to do this with plants in the tropics. And then we started, I now have to say a bit naively, we started to match, again, some big databases, and Matthias is a wizard in data science. among many other things. So we matched splot. I'm sure you hear every second week about that. It's the global vegetation plot database. Milan and Stenka and many others are also very heavily involved. TRI, it's a global database on plant traits. And then again, GLONAV. And we started to match that, and very immediately we realized we have to make at least 1 or 2 years of a detour. And the next 1.5 hours, no, I'm joking, it's not so much. I'm talking about these detours, yeah, which can happen in science, which also I had to learn a lot, and I also try to talk about this next to the science behind that. Why 1.5 years of a detour? So we got, for example, stuck with taxonomic name harmonization issues. We realized that global traits for plants, and specifically alien plants, also have a huge data gap. So first of all, matching those databases was a mess in terms of trait harmonization, which we thought, okay, there's no guideline how to do that. Of course, everyone codes his own little tools, and everyone is having its own solution for that. I was also seeking out to my many colleagues I have, and there was always saying, yeah, I'm doing this, I'm doing this. So there was no real, what is the best way, no guideline. And then we also realized that in terms of traits, going into the tropics is a big issue, because we lack a lot of trait data from the tropics. I have to say, I'm sitting in front of my computer, so I'm not complaining about those who are not measuring enough traits in the tropics. I'm just, saying it's the way it is. And then we also needed to see which plot data we use, and you can't just compare apple and peaches, as we say in Germany. It's actually apple and pears. But you have to find that tropics as not tropics. As a biogeographer, you have a couple of different larger scale tropical biomes there, and then the history of the different tropical regions and blah, blah, blah. So we had also to come up with a set of tropical plots which are easy to work with, which are meaningful to work with, and then having in these plots enough trait data based on the trait availability for the plants in those plots led to a significantly reduced amount of plots. And I'm sure many of you who are doing this type of research know this problem. It's a universal problem, and we were also not surprised of that, but of the whole thing itself. So the first thing we did, how to harmonize the names. Yeah? So that, I said, actually Matthias started and I said, okay, tropical invasions, in 1 year I want to see the paper. I'm off, and you sent me I'm giving you my ORCID number. It was not like this. So we matched these data, and then we realized all of those, for whatever reason, different histories, different people, 3 different taxonomic backbones. Yeah? That means, I don't know if you are aware of that, but those folks, it's me and several others, they used a taxonomic system to say which species has the correct name. Correct is also not true. There's a big discussion what is correct, but which is the accepted name given this system. And they all use different systems. So we had to think about how we harmonize them, and it's not so trivial as you might think. And also because we dealt with tens of thousands of names, you don't want to do this in Excel. Excel is not coming from excellence, yeah? I'm just saying. And also not that I know all of these plant names. I can't always send an email to Milan and say, Milan, can you help me with these 2 species? That doesn't work right. And outside of the taxonomic literature, of course, it's not a new, this is done by hundreds of years by people. There are very few articles discussing this issue, also given this large-scale approach. Yeah? And we thought, okay, why not, writing a guidance and letting other people not going into this rabbit hole we were in the middle. And then in iDiv, I talk a little bit about iDiv. I'm trying to speak even faster. I got a couple of very enthusiastic and great early career researchers. So I'm the old fart here, and they came together and said, yeah, I also have the same issue, or I have skills, can help. And we sit together and thought, okay, why not writing a guideline how to do that best and simulating things and doing it properly, and helping others in macroecology to overcome this issue. And that's what we did. You can also find the paper. Hopefully this is still active, but you can also just ask me. We wrote a paper. What was this paper about? First of all, we described the issue. That's the typical thing what, for example, I'm doing and talking all the time about. So we have 2 data sets just made up. Yeah? let's say one data set is about the conservation status, the other one is the traits, and then you want to mix them, match them. But if you don't have a reference database of the names, if species 1 is also, or species 2 is also species 2, here you get in trouble. And so this is just a depiction of the problem you need. I don't know if I need this. Water. No, I don't even speak into it. Can you hear me? I usually have a loud... okay, sorry. Then I'm speaking more into it. So you see, you have to have the right name matching that species 1 is here species 6, and species 6 refers to this species 6. You get the point, yeah? And if you do this wrong, you just get different numbers. And that might be okay with, you know, one number out of 10,000 is not correct, especially if people argue about what is correct, but you have to solve this issue. And that's very typical. At least in macroecology, people always match different databases or different data sets. Yeah, that's what I said. This is an example. So with harmonization, you have 9 species, and without harmonization, you would think, you don't have to understand this one in detail here, I don't have the time. Without name harmonization, you might end up with 13 species, which is wrong, okay? Just want to make the point here that this is a real issue and not made up just for me, then that I can talk. Right, a good name harmonization reduces the errors. And, but we realized that there's no available overview of tools, taxonomic resources, dependencies among and within those, and there's also no available overview of best practices. And we as macroecologists needed a guide, yeah, and Matthias is also very passionate about open science, which goes along these ways in terms of how to do best practice of data science, and this is one of these issues. And we wrote an overview paper. We identified 50 accessible taxonomic databases. It's not an exhaustive list. We miss a lot, but it was also not our mission. We just wanted to give it a start. And we, for example, selected or depicted those databases according to their traits. Yeah, if they cover global, let's say, the global list of reptiles, or kind of regional, I don't know, the reptiles of Germany, or the reptiles of Saxony-Anhalt, the federal state I live in Germany. And taxonomic breadth might be small here, something like just the toads of Saxony-Anhalt. Yeah, there are just 3. Okay. And then we also then we saw already that many global databases of either small or large taxonomic breadth. What we also did, we did a network of those databases which use what, you know, because you have sometimes a database, let's say the Czech database on plants is used in the European database on plants, and it's also used the information on world flora. So we did also a network of these things that people can understand where my data coming from. And there are sometimes also like GBIF, which is a meta database, and also they started to build a backbone that you can understand where the data are coming from. Very often people are not aware what is the source of my data and cannot understand what is, for example, the taxonomic backbone. Okay? So, and there, as I said, there are a few aggregator databases which sometimes even combine different data sets, which is taxonomically seen not the best way to do it. And those information we gathered also with how many species are in there, is it manually updated, is it updated at all, or is it a dead end? Those data were often not easy to find, so there were one student helper was doing a lot of phone calls and emails, and sometimes even got nightly phone calls because something was a secret or so. It was quite interesting. And we also, of course, because we work, at least I work, and the people around me work with R, we also collected a lot of these packages people use, and we had some criteria, a real R package, you know, not just a collection of scripts, not a wrapper, not being about genomics because we don't feel very confident, we didn't, and still don't do, and also identified in the end over 60, almost 70 packages, of which then we included 60 of. And I'm sure this is now 2 years ago, potentially the number has doubled, which is also an issue. I come to that. And then we identified these packages. That's very descriptive. It gets a little bit more interesting, hopefully a little bit later. We also described those packages because they all do different stuff. which helps people starting to do something like this, to go into our paper. I hope it helps. And then identifying what do I need? Do I need one which accesses databases or wrangling, which usually means cleaning, or is it maybe an infrastructure packages, or can I visualize my data? So we also tried to help here. And this is, of course, the biggest chunk of these packages. And we also did a dependency network of these packages, because sometimes the text size is a very, and I guess many of you have used that, it uses then tool functions of other packages. But you can also see that an enormous amount of these packages are just individual standalone things which might do exactly the same than others. Yeah? And that is something we realized immediately, that people come up just because they love coding, or they hate Googling, or a mixture of both, or whatever, or they developed their own solutions, which doesn't help a lot if you are into open data and maybe want to compare results if they do everything a little bit different. Okay? Yeah, and there's also very little standards in terms of coding, how tools should interact with each other. You know, how much is it allowed to take a function of another packages, etc., etc. And what we did also, and it's still there, hopefully, yeah, it should be, we wrote a Shiny app where you can play around, you can click somewhere, and you can see which database maybe is used by which package, and which databases are dependent to each other, and some of the main traits of these databases. Global, how many species, is it updated, and so on. I think it's a very helpful tool if you start working with larger species data. And this is not just plants. Yeah. And for example, here we see what do the arrows? Yeah, we show, that's exactly what I said. So we see some basic information. We see these different types of arrows and the dependencies among packages and databases. And it's a living database. It still needs to be updated this year, so I have to find someone who can help me with that. And what did we learn? You know, when we actually wanted to analyze tropical invasions, how do we navigate through this landscape? You should never underestimate the amount of work which is in taxonomic harmonization, especially if you work at macroecological scale, and I guess it's also true at other scales when you have to do it manually. You should know the taxonomic landscape. Yeah? There's quite a lot of stuff. It's Wild West, I would call it. And you should also work on your workflows. We realized a lot when we looked into functions that many didn't have record versions, and so on and so on. And of course, also the choice of the solutions depend on your questions. Yeah? I mean, that is something for a calendar every day, another message for your life. Yeah. Take-home message here is that GitHub is not a permanent archive. Yeah? I think it's quite misused at the moment. Rather contribute than rebuild. You have seen we have enough where you can contribute. There's enough tools out there. You don't have to code your own. Use existing expertise. That's also our open science network is very helpful if it comes to tools and linking different knowledge. And if you have to, argue about the have to, build a new tool or taxonomic database, describe and promote it properly. We found some very interesting stuff which just stayed on a GitHub page of a researcher, never been published, never been advertised, but super helpful. Yeah, don't let your work being unseen and unknown. And you should also better characterize the impact of different workflows. We did also in the paper, I didn't talk about this, comparing different workflows, how to match different data sets, and you can come easily to different results. Yeah? And I think this is also true. That's my job. I'm leading a synthesis center. I come to that, and I bring people together. And I think ecologists and taxonomists and database managers and coders, they're all nerds for themselves. I'm totally get this, but they should talk more to each other. Yeah? And that happens, for example, in this community, but it should happen more. And we will work further on completing the network of the resources. Yeah. And so we stopped with the tropical invasions because mainly we couldn't find the right data. And then we realized the second issue. I talked about this, so that's not the second part, but I'm hopefully good in time. So we found a solution more or less or we wrote a guideline on how to do the taxonomic name harmonization. The second issue with these things was then with the traits. Yeah? We work with traits, plant characteristics. And again, Matthias said we are in the tropics, and one of the global data sets is not enough because we have tropical places which have their own data sets on plant traits. So we merged again a couple of data sets. There's BN, there's GIFT, there's TRI, GLONAF, distribution data, trait data, and we did our taxonomic harmonization, and then we wanted to harmonize the traits. And then we realized that we have a lot of species. First of all, again, we harmonize the data, and then we have a lot of species with only one trait. Yeah? So there's a lot of trait data, but usually only the common traits for the common species. That's also not new for you who work with plant traits, but we went out of the temperate hemisphere. We went into the tropics. We were not surprised. It's a super species-rich region. People are still describing new species there. Traits are really hard to measure. Tropical field situations are also very tricky. Everyone who was in the field knows this. So we started to describe the landscape of traits of the global alien flora. Yeah? So Matthias did a brilliant job in harmonizing a couple of trait data sets and also harmonizing trait descriptions. And this is a plot we came up with. So one pixel in this matrix here on this is one trait and one species. And you can see here, was the trait measured, yes or no? What you can see mainly, what it should tell you is that for the large amount of the non-native global flora, we don't have any measured trait. We have quite a lot of species with traits, but given the amount of traits which are somewhere measured, it's quite little. And here you see, for example, a map of the leaf height seed mass spectrum, proportion of alien species with trait combination based on regional occurrences. 100% means all the non-native species in this region have the traits of this trait spectrum. And you can see only, I'm exaggerating, in the species poor, alien species poor regions, you might cover maybe for all of those species, those traits. And again, the tropics, super species rich, we know very little about plant traits of non-native species, okay? And so what we learned here is when working with plant communities and being interested in meaningful functional traits, we still see much relevant data missing. And that's not only plants, of course. We work with plants, but just taking the next step, think about important groups like invertebrates. It looks even worse. Yeah? People then would ask, yeah, what about the beetles in the tropics? Just forget it. You can work with genus or subgenus, and then because it's just so many species. Yeah? Okay, now we realized, okay, name harmonization is not an issue anymore. We found a good pathway, good workflow. Traits, we see that we have an issue here, but we also writing, this has written the paper on the trait gaps and found a good large chunk of data which we can work with, but still realize that we lack a lot of traits and can't work with all the available plot data because they don't have enough trait coverage. And then we went back to the initial idea of the tropical invasions. Yeah? So we really wanted to test what I said already. We wanted to test the taxonomic and functional richness if there's some influence on the invasion pattern. And we used some variables to explain also invasions in the tropics, human influence, some climatic data, and also then some typical data you should use, biome and continent, and the random effect at the plot scale. And we started with very simple GLMMs. And today I show you preliminary results. Yeah, I'm speeding up a bit. And maybe they will change. So in the end, after a lot of subsetting and dealing with a lot of missing traits, we ended with 27,000 tropical full S-plot plots. And then we subsetted them to 12,000 plots. So we lost a lot of plots because they didn't have enough plant trait coverage. We also tried to compare not across biomes, but we used the WWF biomes, that's details. And we only worked at the moment in forests with a known plot data, That's also an S-plot. You lose a lot of data. And these are, in the end, then the plots we can work with. Because we also wanted to have a meaningful trait coverage per plot. So enough knowledge of the traits in a plots of those plants. Yeah? And these are the plots we could analyze. You know, we have 1,200 plots here, starting with 27,000 plots, which are possible, and then we had to apply some data rules, and we ended with 12,000 plots, which might have enough data to analyze what we wanted to do. You lose quite a lot of data in the end. And again, what do we want? What is our hypothesis? The more natives, the less non-natives. Or if a functional level, the higher functional diversity, the lower the functional diversity of the non-natives. Or thinking about a multidimensional trait space, established non-natives have a more distinct functional niche in case like this example here. I can only show the first preliminary results of this one, of these two things here. What did we find? What do we have here? So these are the native herb richness here. In different continents, different colors. I hope you can see this. And this is the non-native herb richness. What we see here is something which we call the rich get richer. It's preliminary. I don't know if it holds true if we go back and increase the trait coverage or using other models. That's what we found with the first analysis. So with increasing native herb and tree species richness, we found the same for trees. Non-native herb and tree species increases too. The hypothesis, the more natives, the less non-natives we had to reject. And this is another hypothesis which is also out there, I can tell you. And we found this one, okay? And on the functional dispersion, one of the examples how to measure functional aspects here, we have here the functional dispersion, native herbs here on this axis, and we have non-native herbs functional dispersion here. And we looked into different levels of human influence. You can see that they are quite distinct from each other. So these lines could be also mathematical artifact. We couldn't find if there is one. So that's why I present this. So what we found is the more functional distinct natives are, the more functional distinct non-natives are too. So quite different to what we actually hypothesized. Human influence seems to increase the functional distinctiveness of the non-natives. So our hypothesis was actually the higher functional diversity of the natives, the lower is the functional diversity of the non-natives, and that's more or less rejected. Even with functional dispersion is one aspect of functional diversity. I don't want to go too much into details. Okay, and just a short wrap up, and then I'm moving slowly to the end. So herbs and trees in the tropics show similar taxonomic and functional patterns. The rich get richer, and the human influence increases functional diversity of non-natives. And our explanatory models were so far not really straightforward, and that's why I'm not showing them. We were not sure. They were not well fitting, and we still have to play around with those. We are still looking for other meaningful and explanatory variables for the tropical invasion. So if you have good ideas, we also have now better ideas. Happy to talk about this. And of course, people come always, why do we see fewer aliens in the tropics? I'm not saying that I know everything. I'm saying I don't know everything. But of course, we thought about this a lot. Invasion history is much shorter in the tropics. Yeah, there's the exploration of those areas has a much shorter history than, for example, in our areas. So there's much less chance to get non-native species by time, at least. And of course, also the research efforts and spending in the tropics is much lower compared to temperate region. That means, by default, on average, less data and also less older data. That influences a lot our data. That's why we are not sure if this is a data artifact, because we don't have the same comprehensive data from temperate regions in the tropics, and that's why we see what we see. Or maybe it's ecology, secondary versus primary forest, etc., etc. So we still have to explore that. Okay. Wrapping up, so biological invasions at large scales I was talking about today. So we have comparably complete data on sub-national static occurrence data of non-native vascular plants and vertebrates globally. It is possible to match different data like distribution data to enrich for example, SDM approaches. I think for GLONASS, for example, that's the next step, because we only have regional data set. We will mix them with plot level data and GBIF, and then calculating downscaled SDM, so better refined distribution data, better than knowing the species occurs in the Czech Republic and Germany, because you can't do ecologically meaningful a lot with that. For some invertebrates, people do this already, because you never will get better data on distribution. And for example, on large-scale biological invasions, microbial invasions are a totally black box. And that's becoming more and more important. There are some microbes which are super nasty, and we transport that all day over the planet. And what is also for biological invasions true, that invasions in less visible ecosystems are still undersurveyed. Soils, deep sea, tropics. And for any other monitoring data, it's the same here. The knowledge on monitoring effort are essential to robustly calculate the rates of change, and it's often also lacking. So I have some slides where we looked into trends and how trends look like if you know about the sampling effort, and they can change considerably. So you have to know how much time was spent in the field to compare, someone who's doing this once to someone who's doing this 10 times. And what are the limitations of the GLONASS type of data? They are, of course, not the holy grail. Mechanistic insights are not possible with regional data such as GLONASS. You can only use this as additional information, maybe adding it to plot data, which happens a lot. And regional species occurrence info don't allow more refined analysis yet. Yeah? You just know it's occurring in a country. And we also saw the limits to it when we were testing some of the hypotheses and GLONASS data, and we really need more fine-scale resolution distribution data of non-native species. They are out there. EVA, the European Vegetation Archive, is a great example of that. But if you want to go beyond that, then there are some limits to it. And some thoughts for discussion. Yeah? So first of all, large-scale invasion research need more fine-grained data, more evenly distributed across the globe. Macroecology is full of great research. Yeah? I'm building my so-called career. I'm not sure about this, but on macroecology, it's always sexy. Yeah, you can publish a global map. That always works. But it's also easy to oversell the power of your data. Yeah? You have seen a lot of global maps which were global and lacking half of the planet, right? I'm sure you do. And also, we often underestimate the workload to harmonize data sets. So I'm seeing a lot of, right now, I'm seeing a lot of applicants who say, in 2 years, I harmonize you the world, and then I'm writing 2 papers. Never. Never. You really have to be very realistic with the workload to harmonize and synthesize large data sets. And the other thing which I had a lot of discussion with Matthias and our center, there are many, many young researchers, is this type of service work. Yeah, Matthias wrote a couple of R packages about functional diversity, for example, and it's a lot of work to maintain them. And we did a service work in writing a guideline. Yeah? So even some people in hiring committees would say a review or a guideline paper is not research. I know, I hear this. But these service work and like creating R packages, harmonizing data sets, publishing data sets, and maintaining these things are very often not similarly weighted like writing a research paper in a journal, right? But that's actually something we have to change. And people like me, and I see a lot of people here who are more senior or hiring people, should also think about what is a service to the community which is also worth to be acknowledged, maybe if you hire someone, because many, many people rely on these service work. Yeah? Some of these R packages have way more citations than I will ever gain, right? And that should be also acknowledged in hiring, that service work for early career researchers is also super important, and not only writing research papers which might get only 5 citations, right? And building careers on such services is even harder. They are very rarely data scientist jobs, purely data scientist jobs. It's getting better, I think, at least in the bubble I live in. And I know many, many early career researchers who would like to build their careers just on something like this and not spending their time in faculty meetings, which I can totally understand. Okay, that was my science part of it. I hope you liked it a bit. Now I was asked, what is the place I'm working at? What kind of place is this? Because it's a nice place, it's a well-funded place. It's not about bragging. It's not my money. It's, if I see it's my taxpayers' money, is a lot, yeah. But I'm not bragging. I just want to introduce the place, which is a great place and offers opportunities for anyone here, maybe to visit or even to work at the place. This is the beautiful building here we have. That was, of course, not yesterday. Yeah, also not in. It's in Leipzig. It's the central of Germany. It's 1.5 hours south of Berlin. For those who don't know where it is. It's called the German Center for Integrative Biodiversity Research. You might have heard iDiv. Yeah, it's like iPhone, iDiv. And these are the 3 main cities the consortium of this project are. So this project, it's a big project, is there since 12 years. So I'm there, I'm the dinosaur of iDiv. I'm there since the first day. And we got funding from the German Research Foundation. It's an excellent center. It has been an excellent center with huge amount of funding. Yeah. And of course, if you give someone 10 million a year, you must be very stupid to not be successful, right? This time is now over. So the DFG funding is over 12 years. It was clear from the beginning we had to apply every 4 years. But now we secured more or less permanent funding. The political situation in Germany is also changing, as you hopefully know. But at least we now have agreed funding for the next 6-10 years. From next year onwards from the federal states. So we have secured funding. iDiv will be still there also after the DFG funding. iDiv is a consortium across 11 institutions. It's not just this building. This is the core house, as we call it. But there are a couple of more institutions which build the iDiv consortium. And of course, we have a lot of third-party projects in iDiv which offer jobs. You know, if we can't offer the job because we have less money now, maybe some of our researchers can. For example, iDiv hosts the European Topic Center on Biodiversity and Ecosystem. This is a big, huge, policy-relevant research initiative or a research center funded by the European Commission. That's the consortium. Just to get an idea, we have the Helmholtz Center of Environmental Research. You might have heard that. And a couple of other centers related to and universities, Halle and Jena. So the consortium is not so interesting for you. This is iDiv. This is maybe a little bit more interesting. So we have research groups like professors like Stenka or Milan. We have a couple of those. I'll show you some of them. We have research platforms like the Jena experiment is something we collaborate a lot with. We have ecotrons, 24 chambers for simulating ecosystems, and of course a lot of databases. I just talked about one. We have a graduate school with, at the moment, I think 80 PhD students and 100 postdocs running through it. We have an internal funding mechanism which even has just calls for internally that you can get a postdoc funded or something to increase collaboration in this consortium. We have a lot of support, other support, and we have the Synthesis Center, which is a place I lead. That's the same, just a bit prettier. I'm sorry for that. And here you can see some faces, and the blue bars are junior groups. These are the professors. And just to give you an idea what topics we cover, macroecology and zoology, theory, experimental interaction ecology, and bioeconomy, so it's an economist here, physiological diversity, conservation, ecosystem services, synthesis, spatial ecology, and evolutionary ecology, symbiont evolution, and so on. We have also quite a strong data support unit. And size-wise, at the moment, we have 300 people in iDiv, so it's quite big. 200 scientists, and 39, or among the scientists, even 52 are international from 41 nations. There's barely anyone who's not, so there's actually, except the housekeeper, the housekeeper person, everyone speaks English. And that doesn't make sense that you have to speak German, for example. Yeah? Everyone speaks a different language. Yeah? I'm the only German speaking in my team, except my assistant. So it's really international, and it's a beautiful place, but it's also a bubble, as you potentially have seen the results of the last election in Germany. And the Synthesis Center, there are many other synthesis centers. You might have heard about ANSYS or CESAP in France. But SDiv is part of this lively research center you see here in the back. And we're trying to have an interaction with the researchers in the center and the external projects we are funding. And what is SDiv funding? And you start to see some opportunities maybe for you. We fund every year, we have calls for different types of projects. We fund, for example, early career synthesis working groups. We fund flexible synthesis projects. We can talk about what this is, but this would be another hour. And we have also quite a lot of postdocs. They either work with working groups or have their own projects. At the moment, 9 of them. And what opportunities does iDiv and SDiv offer in the very end? These are these calls of the synthesis center. So you can just check iDiv.de/SDiv and you find the information of the SDiv calls every year. You can also send me an email. I put you on yet another newsletter email list. We will hopefully also have next year, I guess, a new call for postdocs. And these are not only SDiv postdocs, we are also in iDiv, as I said, the flex pool mechanism. We have very often calls where everyone can apply. We also have calls for the more senior people here who seem to sit in the front here. Sabbaticals, funded sabbaticals, but you are then in Leipzig, yeah? It's not Santa Barbara. And of course, also with 200 scientists, someone is always looking to fill a position. So that is an opportunity, is a nice place, it's an international place. You don't have to speak German. And right now, for example, there's a PhD position in soil science. I just checked yesterday. And there's always someone who looks for a position to be filled. Yeah. We also have a yearly international summer school for master, PhD, and early postdocs, early career postdocs. So there's also an opportunity to just come, it's funded, and to get to know maybe some researchers and also with changing topics, learn about these topics. And I'm happy to talk about anything, not anything, yeah, so not necessarily about GLMMs maybe today. Yeah, that's it from my side. I hope that's correct. And if you are into music, I have to make advertise my own band. Questions?
Yeah, thank you, Martin. So you were so fast. You haven't. So we still have 1 hour. So it will be a huge discussion, I think. Yeah, thank you for introduction, especially of the center, because you offer a lot of opportunities, and I think that our students or researchers could be interested, and it could be a chance for them. So thank you. And now it's time for discussion. If you have any questions, now.
Well, I can scream.
Yeah.
Thank you very much for a nice talk. And you mentioned there's less invasive species in tropics. Does it apply to islands? Thinking, do you have data? Is the difference between tropical islands and the mainland?
Good question. First of all, it's alien species. I'm not sure invasive. I don't talk a lot. Alien, it's a difference. Tropical islands, I don't know. I don't know. I could click now 150 times to get back to the map. I'm not sure if we have a lot of plots on the tropical islands. That's the first issue where I would say, I don't know. So I don't know. My hypothesis would be maybe a bit more aliens, but still also tropical islands. Yeah, no, I don't know. I don't know. We don't have much data on tropical islands.
I would expect that it doesn't apply for tropical islands. And the mainlands, there's not so many incomers like before.
Yeah, the propagule pressure might be less. On the other hand, tropical islands are often tourist hotspots, so I'm not sure about that. And islands itself, usually, if you see it globally, islands have more aliens than mainland. So it could be also on tropical islands. I just don't know. Good question.
Okay, I have a question regarding the Daktarjan regions study. In the analysis, in the second clustering, you included all the native species plus non-native,
Yeah.
right? And so I was quite surprised because non-native aliens, they are not so many. You mentioned just 4%
Yeah.
of species.
Yeah.
And it produced totally different
Clusters.
picture.
Yeah.
So my question is, what would happen? In the native species clustering, you had 7 clusters.
Yeah.
What would happen if you use the second dendrogram and pruned it to 7 regions? Because you could do it.
Yeah.
So,
Yeah.
so if you have 7 clusters or 2 clusters, it just depends on the level
Yes.
of pruning of the dendrogram.
Yes.
So
Yes.
would it be totally different?
Yeah, yes and no. I'm sorry here, but I think it makes more sense if I'm clicking even a bit more. We actually discussed that a lot, and that's, sorry for that. But I will, yeah, let's stick here. This one, right? Yes. And you just said, what would happen if we cut it off somewhere else?
Yes.
Yes. So we played around with we played a lot with the number of realms with this threshold here. In the end, the story stays the same. We can also have a clustering with 8 realms, and with 6 we had this. But the story I want to focus on is that you lose cluster when you add the non-native species. So of course the 7, I would not necessarily say this is a number you should, because it's just a matter of mathematics. There's some sensitivity analysis. We did quite some of them, and we had to find some threshold which can be applied across both dendrograms, right? And you can see that it is not always a straightforward cutoff. Here we cut off here, in this case, yeah, and we could also cut off here. Then it would be one realm less. What we did, for example, also using we did it last week, or the calculation, we also changed the number of aliens in the regions to see if it's an artifact of numbers. Because we have here, if you have, might have 500 aliens out of a flora of 3,500, something like in Germany, and here you might have 50 aliens out of the flora of 16,000. So it's different. We played around with the numbers. The main story, the loss of the realms, the reduction so towards less realms stays the same. So you are right, it could be 9 or 6 if this is the answer. The 7 is in this case just the clustering we found makes most sense. Also actually because there's another group who did the clustering on the native side, the Göttingen folks, because it's their data, the native species, and they used a method where they then said, let's use the 7 realms. And then because we wanted to be comparable to them, we used the same method. But yes, it's an artificial choice that this is 7, more or less. There is some sensitivity behind there.
Yes. And did you also try to do clustering with naturalized species only?
We did this as well, but I can't remember. I have a folder of that. We did as well, but I don't know. I didn't find this too interesting because people did this already partially back in the days. We did this as well. We might put it in the appendix. I can't recall, it's quite a while, how it looked like. But there were some clear clusters, like, for example, New Zealand and Europe, and so because of the same species, you can imagine, right? And also Australia and the Cape were popping up a bit. But because we have quite a lot of cosmopolitan species, then you get a mixed mathematical pattern of that. But that wasn't what I want to focus on. And this paper is meant to be a short paper, and so we didn't want to analyze it until end of the life. But a good question was this one here. I will check if we make sure that this is clear that this is a more or less random choice with some sensitivity in it. Yeah, I guess people would ask for it. If you ask for it, then the reviewer will do the same. So we.
Yeah, but I understand it because you are now asking about homogenization.
Yeah.
Eh?
Yeah.
So you are interested in the homogenization of the whole flora.
Yeah.
So if you had naturalized, then it's more homogenized, and it shows like more similarities between distant regions.
Right.
So it's okay for your question.
Yeah.
I'm just interested in someone else.
Yeah. No, it's a totally fair point. Our model also showed that, of course, distance decay doesn't play so much role because with us overcoming natural biogeography, which is distance decay, this also shows the model that some of these typical rules applying to natural biogeography are just falling apart because of us.
Let's say Mediterranean flora in the European Mediterranean
yeah.
or in California. So these would be on the sub-trial
Yeah.
level, basically. But
So we talk about this region here.
yeah, but I still don't see the similarity. I mean, but this all is based just on species, right?
Yeah.
On the occurrence of species.
Yeah.
No coverage.
No, I wish I would have coverage, yeah.
Of course, some invasive species can cover a lot.
Yeah.
But generally, I mean, what makes the impression on you, but also on herbivores and
Yeah.
so on, is the abundance of different
Yeah.
plants.
Yeah.
And as long as this is not taken into account, it's a very distorted picture of reality. That's
Yeah,
what I
you will not become a reviewer of this paper.
I'm happy I have enough other things.
Yeah, I'm sure. No, you're right. This is just 0, 1. Do you occur there or not, right? And not are you rare or are you covering like Lantana in the Cape region, covering a lot of places. No, the next step, of course, this is a distorted. If you think about abundance or coverage, then you could call it distorted, and it's just based on mathematics. You can also argue about the mathematics like Milan did, but it doesn't take into account in terms of coverage. So rare species more or less have the same weight than very common species, which are, but that's just the data world. But that, yeah, it's a good point, more or less. But this is the typical argument we get as ecologists. It's all good, but your data are shit. I just can't change it, right? But we will discuss, abundance. I can't change it. I mean, we could do some sensitivity analysis. If you would do splot, the full splot data set, which covers almost the whole planet, so it's a plot level based database, and you would kind of upscale. It also doesn't cover all habitats. Yeah.
But these are like floristic regions and not biomes.
Right. I mean, it's macroecology. It's a different concept, but it's a distorted kind of influence you see here because we don't take into account coverage. Yeah? Rare species, a species which occurs only in the harbor of Cape Town, gets the same value more or less like Lantana, which occurs across whole landscapes. Good point. I will take it on the list.
Okay. Michal was the second one.
Sorry to take the time because I have also the follow-up for this one. For me, I mean, the loss of beta diversity is inevitable on the global scale because you're putting species to more spaces.
Yeah.
What I would like to see is when these realms, the lower realms, appear on which level.
Yeah.
So they will appear, definitely.
Yeah.
They are somewhere in the default structure, so that would be something that attracts me to see when they will pop out in different.
When it becomes more diverse. Yeah. Now I'm speaking about things I haven't analyzed with these data, but I also did some analysis on this one. You have to go quite onto a lower spatial scale until you see dissimilarity due to non-native species. And that's by a chance. If you have a small plot, it's very unlikely to see all the plants, but just to step in one of those. So usually we find the opposite pattern of homogenization on very small spatial scales. We just haven't looked into that. So we haven't done this dendrogram until, I don't know, sub-provinces and sub-provinces. We just didn't. It's a lot of calculations. But yes, but I would guess from my knowledge, maybe I'm wrong, but from my homogenization knowledge, is that increasing the dissimilarity you see on very small spatial scales, just for likelihood chance. Yeah.
I think it's also kind of a mathematical artifact that you are putting some strong signal of few species, which are, let's say, everywhere, against something which is very heterogeneous.
Yeah.
So you override it.
Yeah, this is a second argument against macroecology all the time. Yes. I can't change it.
Okay, thank you. So we can continue with other questions.
Come on, grill me.
Jeff, and then.
So I think dealing with biomass is going to be extraordinarily important. So the same idea as coverage. So let's take a look at, say a gradient for the biomass versus richness of native versus non-native landscapes in North America. And I can tell you that when you're in South Florida,
across all taxa, you said snails. Yeah.
Well, and even across, I mean, you're going to also have the non-natives are going to be, and that's what you see in Hawaii, it's what you see in Bermuda, are what have taken over.
Yeah, yeah.
And so if you do it as on just a, you know, a species basis, there's a whole other issue you got to get at. We don't know, you know, do we believe the taxonomists when they tell you that's a species?
Yeah.
You know,
Yeah.
the error rates in land snails, and I don't think land snails are particularly bad. 50 to 80% of the traditional taxonomic categories are wrong.
Yeah.
And it's because they're using a worldview that's based on basically pre-scientific. They're using kind of, you know, they're not using scientific method to describe their species.
Ah, okay.
So the problem is taxonomy is still stuck in the 1400s, and they're happy about it. They don't want to move, and they don't want to modernize.
Yeah.
But because of that, when I start moving to doing macroecological research of land snails, I'm dealing with taxonomic error rates of 50 to 80%.
Sorry.
And I don't think, having grown up as a plant ecologist, I'll tell you, this is not unique to land snails.
No. Yeah. Many good points. You're also becoming on the no reviewer list. You're right. So in some regions, biomass also, of course, I also traveled a little bit and saw quite a lot of non-native species. I don't have the data. I can discuss everything in such a paper, but there are also limits to it. I guess that would be the next step, maybe to look into its basic science. You know, we just describe a pattern.
I'll just say that, like on Bermuda, there's 3 dozen of the 4 dozen species on Bermuda are non-native.
Yeah.
Most of the 17 native species, when you do a survey right now, you're finding across 40 sample plots, chosen for the best quality habitats, singletons.
Yeah.
So what we're looking is, they're still there, but they're barely still there. They almost have blipped out. And in fact, there's been a tsunami of the non-natives. And so it's, you know, and in Florida, you get the same issue.
Yeah.
You know, and having just come back from Costa Rica from the lowland tropics, it's going to be basically the same issue.
Yeah.
That, you know, there's a few invasives that have the highest biomass. In the native stuff, you have to really work hard to find them.
Yep. Yep. Nothing to add. And sorry for the snail taxonomy. I mean, yeah.
Well, like I say, it's not just snails.
But we don't have I would assume working, and I was co-author of one of these global taxonomy backbones, talking a lot and learning a lot from my expert colleagues. The error rates are, as far as I can see it, I'm a zoologist, that's my excuse, is not so high. They are there, you can have 10%, but I would assume with 400,000 species, in this case 280,000 species, even if someone would say, use the, I don't know, WCVP, which we use the Leipzig system, which is the one I was working with, it would not look differently. If this is the right species concept, if someone would, you know, use genetic data for all the plants, coming up with something else, talk to the botanist, not to me.
Well, I know, but I think what you're going to find is that when you start matching actual genetic data to what humans have decided is a species, okay, this is not a land snail issue.
Yep, yep,
And,
yep.
you know, and all I'll say is orchids. If you think, you know, plants are all worked out and the taxonomy is great, orchids.
We had a great discussion about that last week about with the mycologists, because for them it's such a big issue.
Yeah.
So I think we should continue. Yeah. So it's your turn.
Thank you for the talk. This will be something more basic, either something I missed in the talk or I don't remember from my ecology classes.
Okay.
I was really shocked by the number of non-native or invasive species at the islands.
Yeah.
I can't imagine walking on an island and 70% of
Yeah.
the plants are what is a non-native species in this case? How long into the past are we going?
Good question. Sorry, I didn't say this. I assumed you know that. Yeah, maybe I should have said this. So this is a purely arbitrary line which we draw, at least from the school of thought I'm coming from. It's 1500. Okay, it's where globalization more or less started to accelerate. That's how we use it. You know, Columbus messed up America, and then everything started to fall apart. That's a short summary of what we did.
Yeah, Rhoda, and then you. Rhoda was the first
I
one.
wanted to ask you how much time do you spend in the field?
At the weekends or in the office hours? No, unfortunately, I've started as a field ecologist. Now I'm sitting in front of my computer. My job is also not being a scientist, but unfortunately too little. And for example, with the point taken, because I analyze data of others and maybe even criticize them for not taking even more data, blah, blah, blah. With the trade space, lack of trade data, we came up with a couple of things which the community and also us can do. And for example, measure campaigns in botanical gardens, for example. Then you have again a issue of interest-specific variability. Yeah, I'm a macroecologist sitting in front of my computer. At the weekend, I'm happy to be out. Yeah.
Thank you for your talk. My question is motivated by the talk of what is your timing. There was a graph that you showed of the first record of new species.
Yeah.
There were several peaks on there. I'm curious whether we could look at those data and decide what sort of events.
Yeah. Yeah. So. Yes, yes. So first of all, I would not potentially, I know the data, I know Hanno who collected the data. Collected means I scanned the internet for data. I would not pay too much attention to that. That is all species, that means all taxa in this data sets. Crustacea, name it, wherever someone found a list of first occurrences of a non-native species somewhere, okay? And of course, it could be that this one marks, I don't know, I'm very bad in history, someone entering China and being interested in certain species, something like this, knowing what Chinese species were there, and you know, bringing the first time a list of mammals, yeah, and maybe here somewhere New Zealand, bringing all the stuff from Britain, from a garden. So that might be, I don't know. I'm not sure if this is just data-driven artifacts of very well sampled regions with a very well-known data from past. Maybe it's also wars, you know, the first exploration of Australia, whatever. These kind of things are also in here. But if you think about how well distributed data are, when a species occurred for the first time somewhere, these people usually looked like me, longer beard, and so you can imagine where these data come from. So it's, I don't know if there are some, and it's across everything we could find in the internet and in books, whatever. It's not comprehensive. So I don't know if outliers make sense to look in the outliers in a non-comprehensive database.
So have you noticed that?
Maybe was that was the first one, yeah?
Okay, for me it's quite interesting the decline in the last
Yes.
years. Does it mean that the floristic work is not so sexy science as it was? That means we have less publications about this? Or it's really decline of the precision test?
I think it's none of both, I think. So it's, now we're starting to talking like in a church. I don't know. First of all, I think it's just a data, there's a lack of publications, right? And if you have a list of something occurring for the first time on an island, it takes a couple of years until you get the data. I don't think that these type of data getting less sexy, right? The opposite, that more and more people getting back, I'm just making it up. In Costa Rica, never thought about, but going to the herbarium in Costa Rica, actually the same, I'm working with someone in Nigeria and he's doing exactly this, now going back to the Nigerian herbaria and looking into the data and creating a database when the first time someone found a species of which we know is not native in Nigeria based on our artificial data. So I think it's still sexy, but people are starting to explore other data. This is for me rather a lack of publications maybe, and it's also for sure not a more robust management of non-native species. I don't see that at this level. For aquatics, a little bit, yeah, because of some regulations with the ship containers and so on, but on terrestrial islands, I think it's even getting worse.
So just one quick observation. You notice it's all on 50, most of your outliers
Yeah.
are on 17 and 50-year intervals.
Yeah, this is a mathematical artifact because of very often these lists are published in, I don't know when it came, but I know it was in the last 50 years. So it's also not that someone knows, this crab came in 1871 in Japan. Yeah. It's sometimes between 18 and 1850, we know this crab arrived, okay?
So anyhow, to answer your question,
Yeah.
there's something obvious.
Yeah, play around with this data set. It's super cool.
We have a huge discussion in the botanical center. Only the zoologists are discussing now. So, botanists, now it's your choice. You sure?
Is the absolute number of alien species increasing with the number of native species, or is it the relative share of aliens? You showed the graph where it's increasing, and of course, when you have lower native species, a few alien species can already make up a big
Yeah.
amount.
Yeah. So first of all, the number of native species shouldn't increase so much. I mean, it increases in the tropics. Every new flora brings up hundreds of new species. I think that's the answer. So usually, if you get more aliens, then the share is, of course, increasing. Is that the answer to your question? I'm not sure what the.
Can you go back to the?
Which? Well, let me see. Which slide was it? This one? No, wait. This one?
The one with the correlation where you
The what?
the correlation between the alien
Oh, wow.
and
In the tropics, yeah, let's see. It's all good. Yeah, here we are. It's all Stenka's fault. She said I should talk for an hour. This not, and sorry. Yeah, I could also, that's true. But now you know the slides. Now you know this. Don't look at it. This one. So what is the question? Yeah. We haven't looked at this. It would look different. The share, yeah, it's a good point. We could also look at the share. This is just first, showing the richness versus richness. The share in a plot.
You could imagine that in the tropics, where you have such a high species diversity, the actual share of non-natives
Super low. Yeah. But that was not the question we were interested. We are not interested how low it is. But I'll have to check. That's maybe we did this. Yeah. No answer.
Another type of bias because this is based on the plots,
Yeah.
plot levels.
Yeah.
And the plots, I know from Europe that they are sometimes subjectively plotted
Yeah.
the landscape.
Yeah.
And imagine in tropic, I never have been a tropic,
Yeah.
but if there are so few plots, and if I will go there, I will sample as rich as possible the
Right.
plots or something
Yeah.
which is
Yeah, we asked that a couple of database managers, and they, at least for those data we had in the final data sets, which was quite minimal compared to the existing data, they said this is not such a systematic issue.
So can I ask why do we have an order of magnitude difference in the length of the richness? So you only go to 8.
It's a log scale, I guess.
But you go to 60.
Yeah, I'm sorry for being sloppy here with the, I guess it's a log scale. No, that's not, no, that's not log scale. That's just the number of herbs, I guess. That's a good point.
It really goes to 60. It's really 10 times more?
No, I guess that's the wrong, yeah, you are right. I'm sorry to show that, but this potentially mixed the names of the axes here. Yeah, you're right. I'm sorry. Actually, I thought I changed this because the last time I said the same. Yeah, sorry. I have to. Yeah.
So when you studied tropical invasions using SPLOT,
Yeah.
did you use only plots that have both the tree layer and the herb layer? Because in SPLOT from the tropics, there are many plots that only contain the tree layer
Yeah.
But
No herbs there.
there's no information about herb layer.
We tried to we compared only those comparable plots. So if there was only tree data, then we only used the tree data and compared them within a subset of groups. And as I showed also in herb results, if there were herb data, but we didn't mix the herbs and trees, putting them together. We really we looked at trees individually, and if there were herb data, we looked at them individually. And of course, we didn't compare a plot with herb data with a plot where herb data were not monitored. Okay. No. And that's another issue that in the tropics, the type of monitoring is just working differently. Sometimes also not all trees, but just DBH of breast height of 10 centimeters, etc. Yeah.
Will somehow change if we will be working with plants on like lower scales, like subspecies or some genotypes when some plant can be native in like many countries.
Yeah.
It can be like different genotypes. It can be like hidden invasions or hidden alliances.
Yeah, because of the second part of your sentence, we usually don't look at this. It's too complicated. Would it change the pattern? I'm not sure. In terms of numbers, it's even less so, but you just don't have the knowledge about the proper distribution of subspecies level. Yeah, good question. For Europe, maybe you could do this. I'm sure you know the infra-specific levels down to, I don't know, hybrids and formers and whatnot. I don't know. It's potentially too complicated because outside of so well-known regions like US or North America and Europe, very few people deal with infra-specific levels. It's just the sheer amount of biodiversity is so high that potentially, maybe for some little groups, yeah, subgenus somewhere. I don't know. Good question. I just don't know.
I think even the levels for distinguishing what is subspecies and what is species are different on different continents.
Potentially.
So you can do it. So what in Europe we have a few species, in Asia would be just one.
Could be. Yeah. So I once digitized the Russian flora. There was 10 books, and they had in every valley another Artemisia species. So I don't know. Yeah, botanists. Yeah. A good question. I don't know.
So I have probably a common question, perhaps also a bit annoying, but you were talking a lot about the harmonization of the taxonomy.
Yeah.
So what's the potential of artificial intelligence in like finding, because it can be very powerful, I can imagine, in finding synonyms and then matching the databases.
Yeah. It's actually not, I don't give so often talks about this, and AI is not so long around. I can imagine it helps maybe with computational power, that you just, say, ChatGPT, here are the 3 backbones, here's my data set, make it happen. No, I don't use that. Also because these are sometimes opinions of people, right? These taxonomic data sets. So we've written a paper comparing 4 of the major plant global backbones. There was some fighting of the people about what is the right approach to say this is rather a synonym and this is an accepted name. Then AI might help in terms of minimizing the amount of workflows. You just, it's finding, if you give a good prompt, then it might help there. But in the end, you still have to know what you're doing. And some of the big databases just mix different taxonomic backbones, which I learned you shouldn't, because then you mix the different opinions, And but that's why many people say, okay, first of all, I match it with WCVP, and what is left, I match then with LZVP, and then I go, and that you shouldn't do. But those people who are telling then you mix different concepts of taxonomy, they usually don't speak to ecologists or macroecologists, they speak to their own peers, and that's why many macroecologists just, because they can, they just do whatever they want. So it might help at some point.
What's your opinion about the modeling? Because you wrote that there is a huge problem with the quality of the databases.
Yeah.
IDF, for example,
Yeah.
is really for some species I'm working with bats, which is problematic.
Yeah.
And based on that databases, there are prepared some modeling for the climate change and the distribution, the
Yeah.
changes, etc., etc.
Yeah.
This is working with the precise model with quite bad databases. So what does it mean as a result?
Yeah. So I'm not the modeler type of. I'm happy to work with in situ measured data, but sometimes you need to model. That was the part with exchange. We also work with indicators in alien species, and you need to have a robust number of occurrences of which you know which are really true and not false positives and blah blah blah. You have to know the species, right? I mean, I also don't know all the species here, for God's sakes, no. But if you look at some, you need to know the ecology, and if it makes sense, then modeling can help a lot. But you should, for example, GBIF, you should have a good understanding that this is also never comprehensive, has a lack in rare species and in some species, in some regions, and so on and so on. But modeling can help if you do it properly. That's my approach to it, and there are very good modeling techniques nowadays. But you need, of course, if your data underneath that unreliable, then you can forget about the SDM, right? And if you don't know, if you can't evaluate if the underlying data are not robust enough, then you shouldn't calculate an SDM on your own. That's why I'm not doing it. But you're right, it can be very much misleading, and especially also with non-native species, people always argue about it changing its ecological niche when it's coming to a new, and so on and so on. So it's not so easy.
I think we have a time for some last question. If there is no, you can continue.
Maybe I can make it a little bit more difficult then, since it's the last one.
Thanks.
So, like, are you also looking at the, like, you know, for example, climate change, right? Like, so with the introduction of non-native species,
yeah.
if there is any correlation, like later on, if it could lead to change in the, some of the parameters.
Yeah, so the question is if climate change maybe accelerate the impacts of invasions, or if there's something what climate change is doing with invasions along the way. We also looked, of course, we did some climate change projections. For example, what we found is that with increasing climate change, certain areas might become even more alien species. I mean, you don't have to be very smart to come up that areas which maybe now rather cold and ice covered or something will see more invasions. We see this already, you know, in the poles. Other than that, it's potentially a black box. I would assume it opens the floors, for example, here in Central Europe for species which are not yet here and or can't establish. There are, for example, Eichonia crassipes. I heard Eichonia is not Eichonia anymore. I don't know. It's one of the water hyacinth, I think is the English name. It's one of the most invasive species in the world. And we had an occurrence in Germany. It didn't make it over the winter. In 10 years, maybe it gets established. Of course, these kind of things happen. That's simple answer to it. And then the interactions of those things are potentially also quite interesting. It's not getting better, that's for sure. Yeah, in some regions it might get better because the established non-natives maybe don't do especially well if it gets more dry, etc. But then we have other problems than the dying aliens, right?